Bismillah ar-Rahman ar-Rahim. This is the description of the data show histological slides of intestine. The small intestine is about 6 meters long and it follows the same general rule for the digestive tube. As you see here, it has a mucosa, a submucosa, muscularis externa, as well as a serosa, which is the peritoneal covering. Here we have a very specific feature for the small intestine, which is the plica circularis. This plica circularis is circular mucosal fold. It's a fold of the mucosa, which contains a submucosal core of connective tissue, which increases the surface area for the absorption about 3 folds. So remember that the length of the small intestine, which is about 6 meters, the plica circularis are factors increasing the surface area for absorption. The length is about 6 meters, the plica circularis, which is a mucosal circular fold. It is circular and not longitudinal. Take care. It's a circular folds of the mucosa with a submucosal core. Not only mucosa, it is also involves a submucosa. And it increases surface area for about 3 folds. Surface area for absorption, 3 folds. Take care. Here we have also the presence of the villi, which are projections on the surface, and these villi will increase the surface area 10 folds. Also, the cells in this villi, these cells covering the villi, are characterized by having a lot of microvilli on the surface of the cell itself, and these microvilli increase the surface area for absorption about 20 folds. So we have 3 numbers, 3, 10, and 20. 3 for the plica, 10 for the villi, 20 for the microvilli. Finally, in this picture, I want to add that the villi are the projections on the surface of the mucosa, while the crypts are the downward invagination of the mucosa. So these are the crypts, and all these are the villi. Here the crypts are transversely cut. This is according to the plane through which the knife cutting the section passed. So these deep areas are all transverse section and not a longitudinal section in the crypts. All these are the villi, but the deep parts here are transverse section in the crypts. This is a transverse section and this is a longitudinal section in the crypt. And this is, of course, the muscularis mucosa. This is the submucosa sharing in the plica. This is just a diagram to demonstrate that the villi are finger-like projections on the surface of the mucosa, while the crypts are downward invagination of the mucosa, forming this simple tubular gland. So I may be asked about the type of this gland. It is a simple tubular gland. Here it is a demonstration for the circular fold formed by the mucosa and taking part of the submucosa, which is called the plica circularis. So here we have on the other side, we have this is the wall of the intestine, and this is another circular fold here. So these folds are the plica circularis, this and this. Also remember that inside the vellus we have the lymphatic vessel called lacteal, in addition to capillaries for the absorption. Also, we have here in the cells covering this vellus, we have 3 types of cells. Here we have the enterocytes and the goblet cells, in addition also to argintophene cells or enteroendocrine cells. This is a very nice picture demonstrating the vellus, and this is the space between the adjacent villi, 1, 2, 3. Here we have evident two types of cells. The most common is the columnar absorptive cells or enterocytes, and it is provided with a large number of microvilli. These microvilli are demonstrated by this red line here, by the red fine striations here, and these of course increase the absorption by 20 times as we have said before. And they have a specific name which is brush border, like the hairs of a brush. These cells are tall columnar with basal oval nucleus, and you have to remember also, in addition to the light microscope picture, you have to remember the EM picture. The EM picture here, how can you study the EM picture of this cell, which is the most numerous cell present in the villi and the crypt? Here the cell is characterized by the following. The most easy thing to remember is the presence of the microvilli. This is very important. This is number one. Number two, they show junctions. The junctions are present between adjacent cells, and as you see here, you have interdigitations. These interdigitations help to join the cells to each other in a very good condition. And in addition, we have junctions. What type of junctions? We have three types. We have the tight, we have the adherence, and we have the desmosomes. So these cells are tightly joined together to prevent the passage of any material from the lumen to the mucosa. These junctions are the tight junctions, the adherent junction, and the desmosomes. In addition, they have interdigitations, lateral interdigitations. As you see, the side walls are showing interdigitations, which fix also the cells to each other. Then add to that features number two. This is the number two, the junctions or the side walls. Then go to the inside of the cell. You will have all organelles in abundant amount except the lysosomes. Then go to the surface of the cell, which is very important. On the surface, you will have a layer called cell coat. This cell coat or the glycocalyx is rich in enzymes. So we have a well-developed cell coat which is rich in enzymes, and the most important enzyme to remember is the sucrase enzyme, in addition to others. But this is very easy to be remembered if you want to study just one example. We have sucrase, we have isomaltase, we have alkaline phosphatase. Then this is the EM picture of the surface epithelial cells or absorptive columnar cells or enterocytes. Then we will study also the functions. What is the functions of these cells? The functions is the digestion. Digestion, it is the major function here, as the coat contains these digestive enzymes. The enzymes are present in the coat or the glycocalyx. In addition to absorption. Absorption of what? Absorption of the amino acids, the glucose, and monoglycerides with fatty acids. Some of the monoglycerides and fatty acids, which are the products of digestion of the lipids. Now it's time to study the goblet cell. Here is the goblet cell. This is the goblet cell. The site, it is present in the villi as well as in the crypts, but their number increases as we go from the duodenum to the ileum, as the fecal matter starts to be formed and it is hard, need more lubrication, so more goblet cells are present. As regards the LM picture, as you see here, you will find that it is having an expanded upper part and the narrow cylindrical lower part, and the upper part is full of the mucus which dissolves during preparation of the section, while the lower cylindrical part contains the nucleus. The nucleus here is dark and triangular, as you see very clearly in this picture. Triangular dark nucleus in the lower cylindrical part. This is as regards the LM. Then we will talk about the EM, which should be also studied for the exam. Here we have to remove this area. As regards the EM, the goblet cells, here this is the upper expanded part, which is full of the mucus containing granules. So we have here granules which are full of the mucus in the upper part. In the lower part, we have, as we have said, the heterochromatic triangular nucleus, and also we have two organelles which are important for the formation of the mucus, which is glycoprotein. So we have a well-developed rough ER, and of course the assistant or the secretory, which is the Golgi apparatus. So these are the well-developed organelles in the goblet cell for the formation of the mucus, which is glycoprotein. And the Golgi here is supranuclear in position. Here, this is the position of the Golgi, because from this Golgi, the secretions, which are the mucus, are delivered to the upper expanded part of the cell. And of course, it is very clear that the function is a secretion of the mucus for two functions: protection and lubrication. This picture demonstrates the connective tissue core of the villus. It is a typical loose connective tissue that contains the blind end, مقفول, lymph vessel called lacteal for the absorption of end products of lipid digestion, and also in addition to the fenestrated capillaries for absorption of the glucose amino acids, in addition to some of the glycerol and fatty acids. And this is the connective tissue cells. Among the connective tissue cells, you may be asked mention one cell present in this connective tissue. You can easily remember the cells of immunity. You will remember the lymphocyte, its daughter plasma cell, in addition to neutrophils and macrophages. So these are the cells present inside the connective tissue core, and all they are related to the immunity. Also, you may be asked about this label. This label here, it is smooth muscle fibers, spindle in shape with central oval nucleus, and so they are strands of smooth muscle. If you have a label on this spindle-shaped cell, acidophilic with central oval nucleus, so it is smooth muscle fiber. This picture demonstrates the villus. This is the villus, and these are the columnar absorptive cells present in the villus as well as in the crypt, provided with brush border, this area, which is nothing but the numerous microvilli, and this is the goblet cell, the upper expanded part with mucus, and this is the lower part containing the nucleus. Also, here we have this core of connective tissue that contains what? Contains the lacteal, lymph vessel, the fenestrated capillaries, smooth muscle fibers, as well as the immune cells like the neutrophils, lymphocytes, plasma cells, and macrophages. This picture is very nice, demonstrating the goblet cells here. This is the upper expanded part. The contents of the mucus dissolves during preparation of the section, so it appears vacuolated like this, and this is the lower cylindrical part. And these are the columnar absorptive cells, columnar with basal oval, with brush border, and this is the connective tissue core of the vellus, containing, this is a blood or a lymph vessel. This is a lymph vessel lined by simple squamous epithelium, and here we have the immune cells inside inside this connective tissue core. Also in this picture, these are the mucous globules. Very nice picture on in the upper expanded part of the goblet cell, and this is the brush border, and this is the columnar absorptive cells or enterocytes. This is also a very nice picture for the vellus, but it is stained with PAS. The PAS here will stain the goblet cell because it contains this glycoprotein, which is the mucus. So it is stained with the purple color using the PAS stain. So these are the goblet cells. These are the enterocytes, and this is the brush border or the coat. Here also the coat is taking some of the stain. This is the coat. Here we have the connective tissue core. Connective tissue core full of the cells. Also we have sections in capillaries or in lymph vessel, lacteal. Also we have this smooth muscle, spindle with central oval nucleus. Suggest what is the function of these smooth muscle fibers. These smooth muscle fibers can control the length of the vellus. Of course, during the digestion process or during eating, these villi will be very long to increase surface area of absorption, so these muscles are relaxed. While during rest, these muscles are contracted, reducing the length of the villi. This is diagram to demonstrate the structure of the vellus and the crypt. The vellus is an upward prolongation of the mucosa containing this connective tissue core containing the central lacteal and the fenestrated blood capillaries, which are the junctions between arterioles and venules. And here we have the crypt, which is a downward invagination forming simple tubular gland, and it produces enzymes for the digestion as well as mucus for lubrication and protection, in addition to endocrine function by secretion of hormones. Take care here in the picture that the absorption of digested material takes place in these fenestrated capillaries and will pass to this venule to the portal vein to reach the liver at the porta hepatis to supply the liver with the nutrients. Again, this is a very nice picture for the demonstration of the villi projecting on the surface and the crypt invaginating into the connective tissue corium above the muscularis mucosa. Simple tubular gland, this is the intestinal crypt. In this crypt, you can notice these cells at the base of the crypt. Present only at the base of the crypt, this is of course the Pannis cell. So the site is in the base of the crypt. As regards the light microscopic picture, it has a very characteristic picture, which is it is a columnar cell, and it has a basal rounded nucleus, and it has a basal basophilic cytoplasm. The cytoplasm at the base here is basal basophilic cytoplasm, and at the apex we have acidophilic secretory granules, which contain the secretion performed by this cell. As regards the EM picture, it has the same picture as regards the outline, and at the apex, you will have the secretory granules. Here the secretory granules are homogeneous. They are having the same density. They are homogeneous. Secretory granules homogeneous. Plus here the organelles present in protein forming cell, which are the rough ER, the Golgi apparatus, and the mitochondria. Of course, you can expect what is the site of the Golgi, as the granules are present at the apex, so the Golgi will be present in between the nucleus and the apex, so it is a supranuclear position. Before leaving this picture, if you are asked what are these cells, they are the goblet cells, and what are these columnar cells, here they are the enterocytes, and this is the connective tissue core of the villus, and this is the brush border, this red line is the brush border of the enterocytes. Now you will move to the other picture. Here, which is a more high power demonstration for the Pannis cells. Here you can see that these are the Pannis cells with the characteristic LM picture. Basal rounded, basal basophilic cytoplasm, apical acidophilic secretory granules. Of course, this is a blood capillary, 1 cell thick. This is the connective tissue, corium, and this is the muscularis mucosa. These are the goblet cells, which are vacuolated. They are present in the villi as well as in the crypt. Then you may be asked here about the function of these Pannis cells. Pannis cells are responsible for secretion of lysozyme. Lysozymes, these are antibacterial enzymes that cause lysis of the cell wall of the bacteria. So they are responsible for regulation of the normal bacterial flora in the intestine. They have an relation to the innate immune function of the intestine. They secrete lysozyme. Take care, lysozyme, which are enzymes and not lysosomes. This is a picture for the slide in the duodenum. Here you can see the villi and the crypts, simple tubular glands. And in the villi, by the high power, you can see the enterocytes, goblet cells, and in the crypts, you can see enterocytes, goblet cells, and Pannis cells. Here, what makes us diagnose that it is the duodenum? The presence of these mucus glands in the submucosa called Brunner's glands. And then we have the inner circular and outer longitudinal smooth muscle. This is a slide also in the duodenum. You can see the villi projecting on the surface of the mucosa, and these are the crypts. This is a projection and this is invagination. And you can see these white spaces inside, they are the goblet cells. This is the connective tissue corium in between the crypts. This is the muscularis mucosa, and this is the Brunner's glands, which are mucus glands. And then we have musculosa, inner circular, outer longitudinal, and this is the serosa. You can see here a part of another organ, which is actually the pancreas. These are the pancreatic acini, and it is always related to the duodenum and may come with the duodenum in a compound slide. Here in the section of the duodenum, you may be asked about the type of these Brunner's glands. Brunner's glands, it is a compound tubulo-alveolar. Compound tubulo-alveolar. And according to the type of the secretion, it is a mucus glands secreting the mucus. They open where? They open in the base of the crypt. They open in the base of the crypt to add their mucus to the secretion of the crypt. So these are the possible three questions about these Brunner's glands, and they are only present in the duodenum. Of course, you remember that the submucosa in general contains the Meissner plexus of nerves, which regulate the function of the muscularis mucosa, while the airbox is present between the inner circular and outer longitudinal and controls the function of the musculosa. Also, you should remember that the villi here are characterized by being broad and long and leaf-like زي ورقة الشجر. Here in the duodenum, the villi are characterized by being long, broad, and leaf-like. This is a section for the ileum. The ileum is characterized by a very important feature, which is the presence of these Peyer's patches, lymphatic nodules, at the anti-mesenteric border. If this is the attachment of the mesentery, here, so this is the border which is an anti-mesenteric, opposite the border of attachment of the mesentery. So this is a very characteristic difference between the ileum and the duodenum. If you want to remember also, or if you are asked to mention differences between ileum and duodenum, you have also the villi. The villi here are less in number compared to duodenum, and they are long, cylinder, finger-like. But the others are broad, long, leaf-like, and numerous. As regards also the number of the goblet cells here should be more than that of the duodenum. Here we have Peyer's patches, there we have Brunner's glands. This is a very nice picture for the ileum at the region of the Peyer's patches. Here we have the Peyer's patches, which are lymphatic nodules, originating in the connective tissue mucosa and piercing the muscularis mucosa to appear in the submucosa. And this is the rest of the submucosa, the inner circular, outer longitudinal, and this is the lumen. Take care here that these are the crypts. These are the crypts, but these are the villi. Notice that the villi here are very short and maybe absent, but compared to the villi away from the Peyer's patches. So over the patches, the villi are very short, as you see in this picture, and sometimes they are absent. This is a picture also for the ileum, showing the villi. These are the villi which are more superficial in position, and these are the crypts which are deeper in position because they are downward invagination. But these are on the surface. These are the villi. All of these are villi. Notice here the presence of the Peyer's patches, which are lymphatic nodules originating in the connective tissue corium and pierce the muscularis mucosa. Pierce the muscularis mucosa to appear in the submucosa. And they are present, of course, at the antimesenteric border, and this is connective tissue of submucosa, inner circular and outer longitudinal of musculosa. These three pictures to demonstrate the major difference between the three parts of the small intestine. First, this is the duodenum, characterized by the presence of the Brunner's glands. These glands are mucus glands, they are vacuolated, they have lumen, they are pale in color. And of course, we have the villi and the crypts as usual. Here in the ileum, we have also villi, crypts as usual, but in the submucosa, we don't have glands, we don't have mucus glands, we have aggregation of lymphocytes. These lymphocytes are actually appear as the blue dots representing the nuclei of these lymphocytes. So all of these are in the form of the blue dots representing nuclei of the lymphocytes, forming these aggregations, which are blue in color, representing the Peyer's patches. But in case of the duodenum, This is the villi, superficial. These are the crypts, and this is the submucosa having no glands at all. And then we have the musculosa, musculosa, musculosa. So if you see these vacuolated glands, this is the Brunner's glands. If you see the aggregation of the blue dots, this is the Peyer's patches. If no Brunner's glands, no Peyer's patches, this is the jejunum. Duodenum, jejunum, ileum. Don't forget that we have other differences as regards the length, as regards the shape of the villi, as regards the number of the goblet cells here. So we know that the goblet cells are more in the ileum and moderate and less. As regards the villi, here broad, long, leaf-like, numerous, but here long, cylinder, finger-like, and less numerous or least numerous. And this is slightly shorter and tongue-shaped. So this is leaf-shaped. This is leaf-shaped. This is tongue-shaped. And this is finger-shaped. Here are numerous, less, and least. Then we have, of course, anatomical difference, which is the length 25 centimeter. Here we have 2.5 meters. Here we have 3.5 meters. Try now to assess yourself by this trial exam. Here we have the first picture. It is, of course, for the small intestine, as we have these villi and these crypts. Then one will be what? Will be the plica circularis. Two will be the villi projecting on the surface. Three, all of this is the mucosa, ending at the muscularis mucosa. Number four, this is the connective tissue, this is a submucosa that shares in the formation of the plica circularis. Number five, it is lymphoid tissue present in the submucosa. It is the GALT اللي هو gut associated lymphatic tissue. Gut associated lymphatic tissue. If you have an arrow here, these are the crypts. These deep parts here, all these are the crypts. Name the structures in the field. In the field, of course, of view, we have the villi. In the field, we have villi, and also we have crypt. Number two, identify L, C, and MM. L, here, it is the connective tissue of the lamina propria. C, it is crypt of Leberkin. MM, muscularis mucosa. Black arrow, these are the smooth muscles, smooth muscle fibers present in, and also here present inside the villus. Nice question. This is the intestinal villi. Name the cells outlined by the black. By the black, they are the enterocytes and or the absorptive columnar cells, and the blue circle, of course, is the goblet cell. Name the structure outlined by the red circle. It is the villus, and by the black circle, it is the crypt of Leberkin or intestinal crypt. The red star, it is the lamina propria of connective tissue. Lamina propria of the connective tissue, or lamina propria of mucosa. Black star, it is the muscularis mucosa, which will end the mucosa. And after this star, you will have the submucosa. If you have a label under this muscularis mucosa, this will be the submucosa. And name the structures stained positive with BAS. L star, this is the goblet cell. Why? Because they have mucus, which is glycoprotein in nature. Arrow heads, this is the brush border of the enterocytes. that is covered with the glycocalyx containing also the enzymes secreted by the intestinal absorptive cells, which are glycoprotein in nature. So these two locations contain glycogen element or glycogen component in glycoprotein. This is section showing crypt of Leberkin. Identify one. One, this is the goblet cell. Two, this is the connective tissue of the lamina propria of mucosa. Number three, it is in the center of this tube, so it is the lumen of the intestinal crypt of Leberkin. Number four, very characteristic. These are the Pannis cells. Number five, this is the muscularis mucosa. This section shows a lower part of intestinal crypt. Identify the cell marked by the letter A. It is the Pannis cell. Mention its function. If he wants one function, you will mention that it secretes lysozymes, which have an antibacterial function. If he wants two functions, you will add intestinal enzymes. Identify the layer marked by the red line. It is the muscularis mucosa. The encircled structure is mitotic figure. Here we have dividing nucleus. Mitotic figure, then this should be in stem cell. The stem cells, which are undifferentiated cells, are present mainly at the base of each crypt. They undergo division, mitosis, and migrate to replace dead cells in the villi or in the crypts. If you are asked to mention one EM feature for this cell, they have a lot of free ribosomes and polysomes. If you want to mention one LM picture, they show mitotic figures and they have a pale cytoplasm with appearance of columnar cells with basal oval nuclei. Identify the organ, be specific. This means you should mention the exact site in the small intestine, which is the duodenum. Name the structures marked by the black stars. These are the villi. Describe their shape. They are broad leaf-like, broad, long leaf-like. Name the structure marked by the red star. These are the Brunner's glands. Mention the function, secretion of the mucus. This mucus actually contains neutral and alkaline glycoproteins in addition to bicarbonates. Bicarbonates in addition to neutral and alkaline glycoproteins. This makes this secretion is alkaline. It is an alkaline secretion. This alkaline secretion help two functions. Number one, protection of this proximal part of the small intestine by neutralization of the acid-containing chyme coming from the stomach. Number two, it provides the optimum pH for the action of the pancreatic enzymes that are delivered to the duodenum. Identify the organ, be specific. It is the ileum. Why? Of course, because of the presence of these lymphatic nodules, Peyer's patches. Number B, what are the structures pointed to by the black arrow? These are the villi. Yellow, these are the crypts. White, these are the Peyer's patch. Identify the layer marked by the green line. It is the musculosa or the muscularis externa. If this line is only at this area, it is the inner circular. If it involves all this thickness, it is the whole musculosa, inner circular, and outer longitudinal. If it is present only in this area, it is the outer longitudinal. And the surface, of course, will be the serosa. Last question, identify the organ, be specific. It is what? It is the duodenum. طبعا عرفنا from the Brunner's glands. Identify the structures indicated by the green. These are villi. Red, these are crypts. Yellow, this is the muscularis mucosa. Black, these are the Brunner's glands. Black line, this is the musculosa or the muscularis externa. If he is marking only this area, this is the inner circle. This area, it is the outer longitudinal. The organ outlined by the black circle, of course, it is the organ which is closely related to the duodenum, which is the pancreas, that will be studied in full details in the data show of pancreas. This is a diagram showing the part of the wall of the large intestine, starting by the cecum and ascending colon, transverse descending colon. Cecum and the colon have similar structure. Here you will notice that no villi are present, only we have crypts. The crypts here are very deep and wide. And as regards the lining cells, we have the surface epithelial cells can be seen here. As we don't have villi, we have the enterocytes and goblet cells. In the crypts, we have the same two types of cells in addition to enteroendocrine and the stem cells. But take care, no Paneth cells at all in the large intestine. Here is the connective tissue of the lamina propria containing blood vessels and the fibers and the cells. And here we have GALT, gut associated lymphatic tissue. We have this follicle in the lamina propria of the mucosa. Then we have the muscularis mucosa, inner circular, outer longitudinal, and the connective tissue of the submucosa with the blood vessels and nerves of what plexus of the Meissner plexus to control the activity of the muscularis mucosa, as you see in this picture. No glands are present in the submucosa. This is a very important feature, no glands in the submucosa. This is a nice picture for and very characteristic picture for the large intestine, where you can see the crypts in the mucosa. These crypts are very wide and very deep, and the mucosa itself is folded, forming these folds, and these are the spaces in between these folds, spaces of the lumen in between these folds. You see here, this is the mucosal crypts, and then we have the muscularis mucosa and the submucosa here. This is the submucosa, all of this, and then we have the start of the musculosa. This is a transverse section for the large intestine, characterized by the presence of many folds of the mucosa. The mucosa itself is formed of crypts, which are simple tubular glands, and they are wide and very deep. Here is the lumen, and then we have here the muscularis mucosa, and this is the submucosa. Then the musculosa formed of the inner circular layer, and this is the outer longitudinal present in the form of three bands. This is one, second, and third. This is the tinea coli. Again, you can see also here a magnified picture for the tinea coli. This is the inner circular, and this is the submucosa, the mucosa, and this is the outer longitudinal, formed of three bands and not continuous through around the whole circumference of the large intestine. This is a picture demonstrating the fat cells accumulating under the serosa called appendices epiploidy. And this is the wall of the large intestine, and these are the circular folds present in the mucosa. This is a macroscopic picture for the large intestine. As you see here, we have the ascending transverse and the descending column. Here we have the three bands. This is one band, and this is another band, and there is a third one. These bands appear similar to the parasite known as tinea, so they are called tinea, but they are actually the bands of the outer longitudinal muscle fibers, so they are called tinea coli. شكلهم زي شكل الدودة الشريطية اللي هي بتبقى طويلة جدا واسمها تينيا، فعشان كده سموهم تينيا كولاي. But they are actually the outer longitudinal smooth muscle fibers in the musculosa. Here the appendices epiploidy, collection of the fat under the serosa, and these are the circular folds present in the mucosa, very characteristic. This is a drawing for the appendix, which is different in some points from the cecum and the colon. Here also we don't have any villi because it is a part of the large intestine, but the crypts here are irregular. They are actually shorter, and the goblet cells here are not so numerous like that of the large intestine, as the food does not pass through the appendix. In addition to the goblet cells, we have the columnar absorptive cells, stem cells, M cells, and enteroendocrine cells, also no Paneth cells at all. Here the crypts are actually fewer in number. The connective tissue here of the mucosa, which is called lamina propria, contains the lymphoid tissue that pierces the muscularis mucosa, which is ill-developed, cannot be seen clearly, and pass to the submucosa. And here the lymphatic tissue is very well developed, not few follicles, but the follicles are present all through the circumference of the appendix, which has an immunological function. Also, the muscularis externa here is not so developed. It is thin, formed of inner circular and outer longitudinal. The longitudinal is continuous, does not form tinea coli. Also, we have acidosa, but here we don't have an accumulation of fat cells called epididymis epiploidy like that of the cecum and the colon. This is the picture for appendix, which is one of your histological slides, glass slides. And this appendix is of rabbit. Why? Because it has a characteristic inverted Y-shaped crypt. This is a crypt, and it is branched like this, looking like an inverted letter Y. And as you see here, we have the lymphoid tissue well developed in the mucosa and passing to the submucosa, and it is present all through the circumference of this organ. Here again, a transverse section in the appendix, hollow organ, having no villi but only crypts. The crypts here are few and they are short. Here, as you see, they are not so numerous like that of the cecum. And the most important feature is the presence of the lymphoid follicles here in the submucosa and mucosa, and forming a complete circle in this structure, not localized to antimesenteric border like that of the ileum. No, it is spread all through the circumference of the organ. Also here we have these lymphoid follicles that actually start from the mucosa and pierce the muscularis mucosa, which is ill-developed, and appear in the submucosa, and they are present all through the circumference, not localized at one side. Very important to distinguish between the ileum, which has lymphatic tissue, and the appendix, which has also lymphatic tissue in the wall. Here, in case of the ileum, we have villi, which are cylinder-like, looking like fingers, and crypts here, this area, the crypts, and the lymphoid follicles are present only at the antimesenteric border. Here we don't have any villi, we don't have any projections inside. Here we have projections. We have only the crypts. And the lymphoid follicles are present all through the circumference of the organ. Let's start by this first slide in the trial exam. Identify the structures indicated by the black arrows. These are crypts, of course. The white arrows, this is tinea coli. And what is the organ? It is the large intestine, and between two brackets, be more specific and say colon. The second slide, this section is in large intestine, between two brackets, colon. Identify A, crypt of Leberkin or intestinal crypt. B, lamina propria of the mucosa. Black arrow, muscularis mucosa. Black line, submucosa. Slide number 3. Identify this organ, large intestine, colon. Identify the structure indicated by the arrow. Number 38, it is the intestinal crypt. Number 39, Identify the cell indicated by the arrow. This is the goblet cell. As you see, it is empty due to dissolved mucus. 40 here. Identify the layer indicated by the arrow in 40. This is the muscularis mucosa. Slide number 4, this organ is, look at the organ. We don't have any projections here. We have only invaginations, so these are crypts. We have lymphoid tissue all through the circumference of the organ, so it is the appendix. Mention one visible feature, very evident, no villi, only crypts. Lymphatic nodules are all around the circumference of the organ. Inflammation of this organ is known as appendicitis. Appendicitis, appendicitis. Itis means inflammation. So it is an inflammation in the word before it. So it is an inflammation in the appendix. Appendicitis اللي هو التهاب المصران العضلي. Number 5, identify the organs A and B. Identify organ A and B. A, here we have the lymphoid follicles all through the circumference, so it is the appendix. Here you have to mention two criteria: the presence of the lymphatic follicles all through the circumference and the absence of the villi. Here B, it is the small intestine ileum. Why? Because the presence of the villi in addition to the crypts and the presence of the lymphoid follicles, Peyer's patches at one side only, which is the antimesenteric border. So the presence of villi and the Peyer's patches, which are present at the antimesenteric border. Very nice slide. Photos A and B show the basal parts of the intestinal crypts. Right, this is the basal part of the intestinal crypt. Identify each organ. Organ A, it is the small intestine. Why? Because I can diagnose here in the basal part of the crypt the Pannis cells. The Pannis cells which have the apical acidophilic granules and the basal part which contains the rounded nucleus in addition to basophilic cytoplasm. Here on the other side, I cannot see these Pannis cells, and on the other side I have a lot of goblet cells. So the final answer for the differentiation depends on the abundance of the goblet cells and the presence or absence of the Pannis cells. Identify the different organs of the gastrointestinal tract, A and B and C and D. Starting with the A. Here the A, it is lined by non-keratinized stratified squamous epithelium, so it is the esophagus. Number B shows villi as well as crypts and mucus glands in the submucosa, so this is the small intestine duodenum. Here, number C, we have the crypts only. These crypts are deep and they are full of goblet cells, so this is the large intestine column. No villi, deep crypts, numerous goblet cells. Number D, here we have glands, and the deep part of the glands appear to have basophilic cells, and the central part rich in the acidophilic cells, and this is the duct of the gland. Look at the ratio between the duct and the secretory portion. It is 1 2, this is 1 2 3 4 يبقى إذا it is 1 to 4, so this is the fundal mucosa. This is the fundus of the gland of the stomach because of the presence of the gastric pits and the gastric glands with the ratio of 1 to 4 and the presence of the basophilic cells in the lower part of the gland, which is the chief cell or the peptic cell. Finally, you should summarize the identifying features for the different parts we have discussed. As regards the mucosa of the esophagus, it is lined by non-keratinized stratified squamous epithelium. As regards the submucosa, The submucosa of esophagus dot is full of the mucous glands. The muscularis externa formed of inner circular and outer longitudinal. The outermost layer it is adventitia except in the lower part which is covered by serosa. In case of the stomach, the mucosa shows the gastric glands with a pit and secretory part, and the ratio differs from the pylorus to the fundus. As regards the submucosa, we don't have any additional features in the submucosa. It is only the typical connective tissue with the blood vessels, Meissner plexus of nerves, and lymphatics. In case of the muscularis externa, it is different in the fundus from the pylorus. In the fundus, it is 3 layers, inner oblique, middle circular, outer longitudinal, but in case of the pylorus, it is inner circular very thick to form the pyloric sphincter and outer longitudinal. It is covered by serosa. In case of the small intestine, in the mucosa, you will have villi and crypts, in addition to GALT. And in case of the submucosa, you will have glands in the duodenum and Barre's patches in the ileum, nothing in the jejunum. In case of muscularis externa, inner circular and outer longitudinal, and finally we have serosa. In case of the colon and the cecum, it is lined by the surface or the absorptive columnar cells with goblet cells, no villi but only crypts which are very wide and deep and full of the goblet cells. In case of the submucosa, we don't have in the colon additional feature in the submucosa. And in case of muscularis externa, we have inner circular and the outer longitudinal in the form of tinea coli. In case of the serosal covering, it shows the appendices epiploidy. And you can add also the appendix. It is different from the colon in few points as we have said before.